Christmas lectures, number 5, hemoglobin. "20 seconds. Ten, 9, 8, 7, 6, 5, 4, 3.
Well, thank you for that welcome, but I don't deserve it at all, because today's lecture on hemoglobin, the breathing molecule, is going to be given by one of the great pioneer leaders of molecular biology who spent his life working on this remarkable protein. So I have very great pleasure in introducing Dr. Max Perutz.
When I was a student, I wanted to solve a great problem in biochemistry. One day, I set out from my hometown, Vienna, to seek the great sage in Cambridge. And I asked him, how can I solve the riddle of life? And the great sage was John Desmond Bernal, who worked in the Cavendish laboratory where the atom was first split. And he was an X-ray crystallographer who had learned his trade here with Sir William Bragg in the 1920s. So he said to me, the secret of life is in the structure of proteins. And my method, X-ray crystallography, is the only way you can solve it. Now, he said this because 2 years before I arrived, in 1934, he and Dorothy Crowfoot, as she was then, now she's Dorothy Hodgkin, made a great discovery. Before that, people had thought that protein crystals give no X-ray diffraction patterns. Bernal and Dorothy Crowfoot discovered that indeed they did if you kept them wet, because they are sponges, and they got this marvelous X-ray diffraction picture of pepsin with a lovely hexagonal pattern that you can see, and with spots going out to spacings that correspond to the distances between atoms. So what this picture showed was that it is possible in principle to solve the structure of proteins. So when I came to Bernal, nobody had a clue how. And what was worse, when I said to Bernal, well, what sort of protein do you think you could let me work on? He didn't have any. So I met a biochemist and I asked him, what sort of protein could I try my hand on? And he said, why not try hemoglobin? It's the most interesting of molecules. It has a variety of functions. It's the vital protein of the red blood cells. If you can find out how it works, that would be absolutely marvelous. So I asked, well, how can I get hemoglobin crystals? He said, simple. You catch a rat and prick its tail, get out a few drops of blood, and spin off the red cells, wash them, and then you add 1 milliliter of red cells to 0.4 milliliters of water. So we can do that here. And then by adding water, you burst the red blood cells. The hemoglobin comes out and being insoluble in distilled water, it crystallizes. Now, before the lecture, I put crystals made on this way on a slide, and you'll see them on your monitors presently. There they are. So, you see, this is an experiment any of you could do in your school lab, provided, of course, you can catch the rat. What does hemoglobin do? It transports oxygen from the lungs to the tissues, and it helps to take the carbon dioxide from the tissues back to the lungs. But that means it has to be done in the blood. So how does the blood go round? Now, I've designed a simplified model of the heart and of the circulation of the blood, which was constructed by Dave Hart and Chris Rayburn in my lab at Cambridge. And there you see the model. So the heart consists of these two rubber balloons representing the left and the right chamber of the heart. And here you see the body, and there you see the lungs. Now, attached to these balloons are valves. They are consist of little ping pong balls which rest on a ring, and if you press, the ping pong ball rises and liquid can pass through in one direction. But you see, if you press, that ball is pressed down, so this valve is closed. So the valves ensure that blood can flow this way through, and again, when you press here like so, it can only flow that way through. Now, if somebody would like to come out, would you, and work this machine? Yes, right, come along. What's your name? Matthew. Well, look, would you like to hide behind the back of the machine? And then, look, wait a sec, let me show you. Press each balloon hard, you see, and press it right down until you have emptied it all, and then count 1, 2, 3, and then press the other one slowly. Okay, so if you go to the back and do this from the back, you see, so I said this is the left, that's the right chamber of the heart. So we are looking at actually at the back of the person in this diagram. All right, you start with this one. You see, you press it hard, and you see when he presses, this valve opens, that closes, and that one opens. Now release that. Press this valve opens, that closes, and that one opens. Now do it again. This opens, that closes. So you see, as he, now stop for a moment. You see, as he presses as the left chamber of the heart contracts, blood is pumped through the body and returns to the right chamber. When he presses the right chamber, all right, do it now, then blood is pumped through the lungs and returns to the left chamber. Now, the blood that comes from the lungs is scarlet, and so it's the blood that's pumped to the body, but the blood that returns to the heart is bluish in color, and so is the blood that returns to the lungs. Now, do you remember who first showed that the blood circulates in this way? William Harvey. And when? Yes, about the middle of the 17th century. Thank you very much. I nearly forgot about you and left you. When he proposed that the blood circulates in this way, and he published his book about circulation of the blood, everybody was against it. They came down on him like a ton of bricks. And one of the 100 objections that were made was it couldn't circulate because the arterial blood is red and the venous blood is blue, and how could they be the same? But then Harvey put some venous blood in a dish and left it standing there and showed that when it's exposed to air, it turns red. Now, why does it turn red? Harvey couldn't understand it because, of course, he didn't know what oxygen was. He had no idea So he didn't know even what the purpose was of the circulation of the blood. And that wasn't solved until the great French chemist Lavoisier at the end of the 18th century showed that the blood turns scarlet when it takes up oxygen, and bluish when it loses oxygen. So here then we can put the label in that when the blood goes into the lungs, oxygen goes in, and then when it goes into the body, the oxygen gets out and is used to burn food in the way that David Phillips explained to you in the other lectures. Now the food burns to what? To carbon dioxide and water. So the carbon dioxide has got to get back to the lungs and be exhaled. So now the carbon dioxide here gets into the blood, and in the lungs it is exhaled. It gets out there. So how is this done? Oxygen and carbon dioxide are both reasonably soluble in water. So you might think there isn't really any need for anything but water to be present in the blood. But it turns out they're not nearly soluble enough. If your blood contained just water, you couldn't get enough oxygen even to keep you alive, even lying still in bed, let alone when you move. And without and the carbon dioxide would turn your blood into soda water. So hemoglobin helps to carry them. It increases the quantity of oxygen that can be carried in the blood 15-fold over what could be carried by pure water. And it helps the transport of carbon dioxide by making it soluble in a very subtle, cunning way, which I shall try to explain to you in the lecture. So now, what is this stuff? What is hemoglobin made of? Well, when I started this research, which is a long time ago, before you were born, 1937, when I started it, people only knew that hemoglobin was made of protein, and the protein consisted of about 10,000 atoms of carbon and oxygen and hydrogen and nitrogen and sulfur. And then in addition, of course, there's the stuff that makes blood red. And the structure of that had just been worked out by a chemist in Munich, Hans Fischer, who showed that the red stuff of our blood consists of a ring of carbon and nitrogen atoms like so, with an atom of iron in the center. And it's the iron that is the vital part. You remember, when you're anemic, you're short of red blood cells, you're short of hemoglobin, and your doctor then prescribes iron sulfate. So you eat that, and that gives you more iron atoms, and that in turn stimulates your bone marrow to make more hemoglobin and more red cells. So here's the atom of iron, and as you see, this is the vital part, because this is where the oxygen hooks on. So if you take a molecule of oxygen, it hooks on like that, and you'll be surprised, when you're young, you think always everything is already known, but it wasn't until last year that anybody actually saw this oxygen. Before that, we've always inferred there's an oxygen there. And then my colleague Simon Phillips showed by X-ray crystallography that this is how the oxygen sits at the iron atom, and we could see it for the first time. And that was a great thrill. Now, yes, I didn't tell you what the red blood cells look like. You see, here they are. They look a bit like UFOs, only smaller. They're about the thousandth of the diameter of the pupil of your eye, and they contain just under 300 million hemoglobin molecules each. And if you prick your finger and get out a small drop of blood, that contains about 300 million red blood cells. So that drop of blood contains 300 million squared hemoglobin molecules. Now, I've said nothing about the protein part. We'll talk about, well, we might as well talk about it now. As I said, when I started, people said it was just globin, you see, and globin was a name. We didn't know what it was. But then chemists in Munich and in New York worked out that it is made of 4 protein chains like this, of the same kind that David Phillips showed you before. And here I've hung up one of these chains, which consists of 146 links. And so there are 2 chains like that, and then there are 2 chains which are 5 amino acids shorter, so 141 each. So in one hemoglobin molecule, then there are 4 chains, and there are 4 hemes. 4 hemes, 4 atoms of iron. Right. So now chemists found this very odd. Usually when oxygen combines with iron, it turns it into rust. And then the oxygen can't get off. But so they asked themselves, how is it that the oxygen in hemoglobin can get off again? It goes onto the iron in the lungs, and it comes off the iron in the tissues. What sort of a reaction is that? Now, and they began to measure things. Now, have I got my little cuvettes here? Yes. So I want to show you some of the experiments which people did to find out how that reaction worked. Well, first of all, let me show you these different colors. So here I've got these little cuvettes, you know, just little square vessels with flat faces so you can look through them. And there on your left, I've got hemoglobin with oxygen, and on your right, hemoglobin without oxygen. And you see that this is bluish and that is scarlet. So hemoglobin changes its color when it combines with oxygen, and we can exploit this change of color to measure how much oxygen has actually gone in. And we do that by looking at the spectrum of a hemoglobin solution. So here I've got another cuvette filled with oxygen-free hemoglobin, and it's sealed to the cylindrical vessel so that you can let air or oxygen or other gases into the vessel and shake up the hemoglobin solution with it to get these gases to react with the hemoglobin solution. Now let's look at the spectrum, please. Shall we? I'll hold this out of the way, shall I? So here you see a rainbow type spectrum, and now Bill Coates will put the hemoglobin solution in, and you see there's a dark band here, a single dark band in the green, where a solution of oxygen-free hemoglobin, deoxyhemoglobin, absorbs the light. And now, Bill, if you open the rubber tube and let the air in, shake it up with air, you'll see that this spectrum changes in a very striking way. I should think that's done it, you know. Oh, yes. Now you see 2 bands instead of 1. You see one in the green and another in the yellow. Thank you very much. And now I'll show you this same band in another way. Here we have a lovely machine which actually plots the amount of light that is absorbed at different parts of the spectrum. So here you have your rainbow-like spectrum. You see it here from the blue to the green to the yellow and to the red. And then you will see on this tiny little television screen, you will see coming up a curve that tells us how much light is absorbed at each part of this spectrum. So now, Alan, if you will make this Hewlett-Packard spectrophotometer, that is its official title, work. It's got a built-in computer, and it shows us the spectrum in a flash on this little television screen from where it's shot up to the, you look up on the monitors. There you see a curve, and you see the curve with the two peaks, which correspond to the two bands you saw on the card here. Now, what I want to show you is that we can pump the oxygen off again. So I'll connect this piece of rubber tubing to a vacuum pump, and and open the clamp, and let the hemoglobin solution flow into my vessel, but in such a way that it isn't sucked away by the vacuum pump. This is very important. And shake it nicely. Perhaps until it stops bubbling. I think it needs a bit more. It's nearly done it. All right, now I'll clamp the rubber tubing off again. And let it run back into the cuvette. And now, Alan, if you'd like to show the spectrum again. Let's watch. Ah, it's worked. See, there's the single-banded spectrum back again that you saw of the oxygen-free solution. So, as I said, you can use this. This method to work out how many of the iron atoms in such a solution have taken up oxygen, because if you sort of inject oxygen gradually and measure the spectrum at each step, you can then work out how much of the spectrum is due to the double-banded one and how much is due to the single-banded one, and that gives you the answer. So a group of Danish physiologists did this kind of experiment in 1904, a long time ago, and of course they expected confidently that at the start, when there was no oxygen in, the hemoglobin would have the highest affinity for oxygen. Obviously, you see, when all the oxygen combining sites are free, then you would expect the affinity to be greatest. And then as they gradually get filled, the affinity would fall off. But instead, they found a quite extraordinary property. They found that the hunger of hemoglobin for oxygen increased with the eating. To start with, when no oxygen was there, its appetite for oxygen was small. And then, sort of gradually, it rose. And what this meant is a strange kind of behavior if you think of it in single molecules, which they couldn't, of course. You see, they were terribly handicapped because they didn't know yet what I told you, that each hemoglobin molecule has 4 atoms of iron. But so what I'm telling you now is how we would see the experiment of these 3 Danish scientists, these Danish physiologists, who were called Bohr, Christian Bohr, the father of the great physicist Niels Bohr, and Hasselbalch and Krogh. So, see, supposing these were 2 molecules of hemoglobin, each with 4 hemes, and now this one, say, has 3 atoms. Oh, come on, stay there. This has 3 molecules of oxygen already bound, and that one here has only 1 oxygen. And now we ask ourselves, when a fifth molecule of oxygen comes along, where will it go? To the rich one that's got 3 already, or the poor one that has only 1? And the answer is, what do you think? The rich one, that's right. It'll go to the rich one. So this is where it'll go. And then you see, if you have this kind of situation, and the blood arrives in the tissues, and here's one rich molecule and the poor one, which is the more likely to lose a molecule of oxygen? Is it the rich or the poor? And what do you think? The poor, you see. So the rule of the game is the cruel one, for he that hath, to him shall be given, and he that hath not, from him shall be taken even that which he hath. Now, why? Why do you think it works like that? See, it's immoral, but it has a great biological advantage, because you see the fact that this is the one that's liable to lose its only possession means that when the hemoglobin molecule arrives in the tissues, it will unload all its oxygen molecules. You see, and the fact that this molecule is likely to go here rather than there means that in the lungs, each hemoglobin molecule is going to be loaded up fully with oxygen. So what it means is that it's like a very efficient mechanical shovel, each molecule. It loads itself up fully with oxygen in the lungs, and it unloads itself efficiently in the tissues. So it makes great biological sense, but chemically it makes absolute nonsense. It was one of the greatest puzzles of biochemistry, this phenomenon which is called heme-heme interaction, because you ask yourself, how does this one know that these others have got oxygen already? There isn't a telephone between them. There are no electric wires in protein molecules. And you see, protein molecules are not electric conductors. They are insulators. So there's no way a signal can pass between these. So it was an absolute riddle. And of course, I hoped to solve this riddle in my PhD thesis. The lucky thing was that my examiners didn't insist on it, else I would have had to remain a PhD student for, what was it, 33 years. Yes. So this is one of the riddles. Now, you see, I'll show you another. You see, there's this rule, carbon dioxide out, oxygen in, carbon dioxide out, and the other way around. And now the same physiologists ask themselves, does the presence of carbon dioxide actually affect the binding of oxygen by hemoglobin? And so I'll show you another experiment here. Again, I've got a deoxygenated solution of hemoglobin. So we'll have a look at this. Take this out. Look at its spectrum. So would you like to show this? And so it's the, where is it? Oh, yes. It's the same single-banded spectrum again. And now, it's a tricky experiment. I hope it'll work. I'll inject a little oxygen, 3 milliliters, into this cylinder, which is filled with nitrogen. So, actually, I must be careful. It's filled with nitrogen under pressure, so it might push the piston of my syringe out. All right, so now I'm pushing 1, 2, 3 milliliters of oxygen in and pull my syringe out again. Now I'll let the hemoglobin again flow into the cylinder and shake it up. With this small amount of oxygen I have injected, and do this vigorously. Let's give it a bit more time this time, you see, because I've given it rather little oxygen, so it'll take a while for it to absorb it all. And because of heme-heme interaction, you see the affinity for oxygen at the start is not very great. So let's hope it worked. Would you like to show the spectrum again, Alan? Yes. You see now that if you look closely at the spectrum, it doesn't consist of a single band anymore. It actually shows just the trace of the two bands of the oxyhemoglobin come up. You see not one hump, but two. Now, where is the, oh, here it is. Let's see what happens if I inject carbon dioxide. Again, I must be careful. So I inject 20 milliliters of carbon dioxide. Let the hemoglobin out. Come on. Shake it up. Let's look at the spectrum. I may have done it too fast, but yes, I did a bit. Let's do it once more. Right, let's try again. Not very well. When it worked, it hasn't worked too well. When it this morning, most of the oxygen was expelled by the carbon dioxide, but it's tricky to get it just right. So what these Danes observed was that carbon dioxide expels oxygen and vice versa. And that seemed very odd, because carbon dioxide doesn't react directly with the iron. So they wondered whether perhaps it was, or rather an English physiologist, J.S. Haldane, wondered whether it wasn't perhaps the acid that is produced when you dissolve carbon dioxide in water, the carbonic acid, that sets free the oxygen, and conversely, whether acid is perhaps set free when oxygen is taken up by hemoglobin. So I'm now going to show you that effect. I've got a solution of oxygen-free hemoglobin in here, and I've got nitrogen bubbling over it, and dipping into the hemoglobin solution is a pH meter. And it shows you that the pH is about 7.6, which means that the solution is slightly alkaline. Now I'll stop the nitrogen. Turn this round. Let in oxygen. And then we'll watch what happens. Just a moment. Is it going the right way? Or have I? It's not. Ah, I've not turned the tap the right way, so nothing was happening. Now it should be okay. So now you see the needle is drifting to the left. So, which means that the pH is dropping, which means that hydrogen ions are set free in the hemoglobin solution when it takes up oxygen. So what's the sense of that? Why should it want to do that? Well, the reason is for this effect is that the blood doesn't want to transport soda water. So I need a helper again to show you this effect, which let's see, who hasn't been yet. Would you like to come? Yes, look, so let me first show you what would happen in the tissues. Would you first like to put soda water into this flask? But just a moment, come around here so that people can see what you're doing. If you do it here, yes. That's right. So put it first into this flask. And you see it frothes and bubbles. And now in that flask, I put a little sodium hydroxide. Now put it into that one, into this one. Put it in fast. Stop. And you see it's killed the fizz. So the hemoglobin does the same thing in the tissues. It mops up the hydrogen ions set free when carbon dioxide dissolves in water. So it kills the fizz. It turns carbon dioxide into soluble bicarbonate, which can be transported back to the lungs. And now would you like to pour some hydrochloric acid into here and show what happens in the lungs? Well, sort of up to there, you know, quite a generous helping. Go on. Yes, more. That's it. Can you? So again, perhaps I'll hold it up. Do it again, and pour in quite a lot. There, you see. So this is what hemoglobin does in the lungs. It adds acid. It adds hydrogen ions, which come off the hemoglobin on combining with oxygen. So it frees the carbon dioxide, which can then be exhaled. Right. Now, let me show you another. Thank you very much. When our gas was still coal gas, many people got poisoned each year because they left the gas tap on by mistake and didn't notice the gas escaping, and they got poisoned by the carbon monoxide that was in the coal gas. And why? The reason is that carbon monoxide combines with hemoglobin in the same place where the iron combines. So here we have a model of carbon monoxide, just one carbon and one oxygen, and that goes on to the same place where the oxygen combines, only it sticks 250 times harder. So once it's combined, it takes a long time for the carbon monoxide to come off. Now, there are other things that produce carbon monoxide, car exhausts and cigarette smoke. So let's see what happens if we let this hemoglobin solution here smoke a cigarette. The solution is under vacuum. Again, you see, so when Mr. Kurtz opens the tap, then the smoke should be drawn in. Oh, it wasn't, was it? I think we'll have to evacuate again.
I think some gone in. Some's gone in.
Yes, but not. I don't think enough, really. Can you keep it going? Let's evacuate once more. It looks as though. Right. Now, would you like to try? What about your holder?
Could you turn the... I could just turn it up.
All right.
Got it.
Good. So now this tonometer is fairly... There's a fair amount of cigarette smoke in it. And we'll see what happens now. Shake it up again. Now let's look at the spectrum. It looks right. The solution looks pink. You see, while hemoglobin combined with oxygen is scarlet, combined with carbon monoxide, it's pink. And so people who have been poisoned by carbon monoxide look feverish, which often leads to their poisoning being misdiagnosed. Now, let's stick this in and look at the spectrum. Spectrum looks similar to that of the oxyhemoglobin, only that the bands are in a slightly different position. Could you come and pick this up for me, please? And thank you. Now let's see whether we can pump the carbon monoxide off. Oh, that's enough. Let's try again. There, it's still there. You still see the double-banded peaks. And this means that if you're a smoker and inhale, then some of your iron atoms will be permanently blocked with carbon monoxide, and they won't be available for transporting oxygen. So in a heavy smoker, perhaps up to 15% of the iron atoms are so blocked. And that means, you know, young people like you always say, well, if I smoke, I don't care whether I die of cancer at 60. But the point is that you're more unfit here and now because your heart has to work harder to compensate for the number of iron atoms that are not available to transport oxygen to your tissues when you walk or run. Now, some of the oxygen actually does come off, some of the carbon monoxide does come off, and can be detected by this kind of breath analyzer. And so we would like to show you the difference in the carbon monoxide content of the air in your lungs in a smoker and a non-smoker. So first, may I have a volunteer? Would you like to come out? And sort of hold your breath for a few seconds and then breathe into this bag.
No. Keep going. Keep going. More. That's fine.
That's it. Lovely. So now we'll let a little out. All right?
Right.
And plug it in here. And now squeeze it out. And the scale on here is in parts carbon monoxide per million. And what's your name?
Robin.
In Robin, there are about 4 parts per million of carbon monoxide, some of which come from the environment, and some come from carbon monoxide that is actually produced in the body by the breakdown of heme. Now, let's repeat the experiment with Bill Coates, and see what the result is.
Okay. I want this.
Yes, right? And
Could you hold that, man?
yes. Okay.
Here we go.
Right. Now will you breathe in? He's got to hold his breath. All right.
Okay.
Now let some of it out.
Okay.
Now watch it.
Oh.
So, in the air exhaled by Bill Coates, there are 18 parts of carbon monoxide per million.
That's great.
Now, as I told you, there was I supposed to solve the riddle. But I didn't know how, because, see, if we look at an X-ray picture of hemoglobin, where is it? Thank you. You see, I got these lovely pictures of hemoglobin when I was a research student, and I showed them to all my friends and said, aren't they marvelous? So they said, yes, yes, very nice. And so then they would say, well, what do they mean? And when they asked me that, I would change the subject, because I had no idea. And the reason why I had no idea was that there was no way of working out the way the different waves corresponding to the different spots have to be superimposed in the way David Phillips told you in his lecture on X-ray analysis. So I tried all sorts of ways of solving that problem, but it took me 16 years before I found one that worked. And then it still took another 6 years to put it into practice. So it was 22 years from the time when I started until the time I could actually see for the first time the hemoglobin molecule. And that was a fantastic thrill. I mean, imagine you wanting to know what this thing looks like that's inside the red blood cells and how it works, and everybody asking you. And of course, most of the other crystallographers thought that I'd never solved the problem anyway, and that I was crazy to try a problem so difficult, because when I started, nobody knew the structure of ordinary sugar, and there was I, such a fool, to come along wanting to solve a structure with 10,000 atoms. So, where is it? Ah, here we are. So, there it was at last, this molecule, and you could see the way the 4 protein chains were folded, starting here and going down and forming a complicated kind of loop, coming up there, then going round inside the molecule to form a kind of hairpin bend and ending there. And all 4 chains were folded in a very similar way, although there were slight differences between the black ones and the white ones. And there were the 4 hemes on the surface of the molecule. Now, so that appeared 21 years ago now. And of course, I was wildly excited. You see, I felt as though I had discovered a new continent. And I thought everything, all these questions which I told you about, they would be solved straight away. But again, the same sort of thing happened. You see, my friends came to admire my marvelous model, and then they would ask, well, what does it mean? How does it work? And I didn't have a clue. But imagine this situation went on for another 11 years. And only then, at last, did everything fall out in that most exciting way that you find in protein crystallography. The answers come out on a single day. You build your model and you look at it, and there you see, just in the same way as David Phillips showed, he understood how lysozyme splits the sugars in the bacterial wall, just in the same way. On one single day, looking at the atomic models of my hemoglobin molecules, all the answers fell out. And I knew how these hemes telephone to each other. And I knew why hydrogen ions expel the oxygen and the other way around. And the answer was that hemoglobin changes its structure as it takes up oxygen. So there was quite a big team of us who were getting out the answer. There was Anne Cullis and Michael Rossmann and Hilary Muirhead and Tony North here at the Royal Institution, and several others who slaved away at this work until finally Bill Bolton, I forgot, until we saw that it does a kind of rock and roll dance. You see, if I hold it like this, you see, holding one white and one black chain in each hand, then these pairs of chain swivel relative to each other like so. So it opens up, closes, opens up, closes, and so on. You see, and this is why I called the lecture Hemoglobin, the breathing molecule, because you see, just as your heart goes thump, thump, thump inside you, so these little hemoglobin molecules in your red cells go click, 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 clicking from the venous form, in which the black chains are widely separated, to the arterial form, in which they are close together. So it goes together and opens up and so on. Now, you see, you might say, so what? But that was the first time that anybody discovered a molecule that changes its structure in response to a chemical stimulus. And there are hundreds of other such protein molecules on which we depend, which do the same thing. You know, for instance, if a nerve impulse runs along one of your nerve fibers, then it changes some molecules, protein molecules in the nerve, change their structure, and they let sodium into the nerve and let potassium out, and so on. So this then is not just a unique thing, it is an example of the way many protein molecules work. Now, you might ask, if hemoglobin is a mechanism, you know, if it's not a sort of oxygen tank, if it's a moving mechanism, does it ever go wrong? And what do you do about it? Now, some, how long ago is it now? 40 years ago, I think it was a British medical officer who first found in Africa a peculiar form of anemia. That is a peculiar form of a disease where children were short of red cells, they were short of hemoglobin. And what was peculiar about this anemia were several things. First of all, it was inherited. And it was inherited in such a way that the carriers of this illness were healthy, but if two carriers married, then a quarter of their children would get the disease. And the other peculiar thing about it was that in these sickle cell children, the red cells had their shape changed in a strange way. Now let me show you a picture of such a sickle cell child. There you see it. It has a raised forehead. And that is because it loses hemoglobin so fast that the bone marrow is trying to catch up. And so much bone marrow is made in that the forehead, when it's still soft, is raised up in that strange, unnatural way. Now, can we look at the red cells? These are normal cells. These are the normal cells. Now you see, up there you see a picture of normal red cells, which are saucer-shaped. And then we'll show you the sickled cells by Dr. Porter, Sir George's son, at University College Hospital this morning. And they are drawn out into this strange kind of sausages, sickles, you see, but only in the absence of oxygen. When you put the oxygen back, they go back to their normal shape. So that was an extraordinary thing. And what was it due to? Now, it is due to a mistake in the gene that codes for this chain in the hemoglobin molecule. So here you see the beginning of the genetic message for the beta chain of hemoglobin. And there the sixth residue in the chain is coded for by GAG, and the amino acid is what David Phillips called E, which stands for glutamic acid. Now, Vernon Ingram, one of my colleagues at Cambridge, then a young biochemist, made the startling discovery that in these sickle cell children, the glutamic acid is replaced by a valine. And then later one found that this happens because the A in this position is replaced by a U. So let's look. Can I have the model back now? What does this mean? Look, here's this 6th amino acid, and here's this glutamic acid with the water-attracting oxygens there. All that happens in sickle cell anemia, I'll cheat and take this one off and put it there. All that happens in sickle cell anemia is that these red atoms, these oxygens, are replaced by a methyl group, a carbon, and this tiny change produces the disease. So imagine if one little thing like that, and you'll be ill for life. And why does it do that? The reason is that when this is changed, the hemoglobin molecules aggregate into these strings of pearls, and the strings of pearls then stick together and form fibers like that, and these fibers grow in your red cells like bamboo shoots and draw them out into these peculiar shapes. So what can we do about it? Try and find some way of preventing this happening now that the structure of the hemoglobin molecule is known. Many people are trying, but it's very hard, and I don't know whether they will succeed. Thank you.